Hi, welcome back to InnoFX Live. Today we're going to be talking about implementing the new package integrity testing guidelines. And we have an expanded team with us as a change up in your program. We've gone from Justine Young, we got Jim Veale, Oliver Stauffer, did I get that right?
Yes.
And Don Singer, okay? So, we'll start off real simple. So, what integrity guidelines are we talking about here?
So essentially today we want to be discussing the revisions to the USP 1207 chapter, which is the chapter for sterile product packaging, package leak testing. So what is package integrity? Basically it's the ability of your package to keep the product in and any detrimental contaminants out. For parental products, that is of course microbial ingress. But say, for example, if you have an oxygen-sensitive product, you also want to ensure that you're keeping out ambient air and other gases.
So what other types of dosage forms does it apply to? Just the injectable multi-use syringe, multi-use vials, or?
Any parenteral product packaging, so vials, cartridges, syringes, auto-injectors.
Ampules?
What? Ampules, IV bags, foil bags, non-porous packaging, porous packaging, flexible, rigid.
Okay. So Oliver, how you go about testing this stuff?
I mean, there are a variety of methods, and we're an equipment manufacturer, so we're developing these test methods for the field and the market. But there are a variety of methods out there to test packaging for container closure integrity. Whether it's treating it with a certain vacuum and monitoring that vacuum level, measuring the head space, applying voltages to measure throughput current. So there are a variety of methods that you can use. The most important thing is that you use a method that's reliable and repeatable, that gives you some sort of quantitative, measurable result in the end. And I think that's really where Justine was just starting to hit on, is that the new regulations, USP 1207, that's really what the meat of it is driving the industry towards the next generation of test methods and methods that are more reliable.
So what method, so what do you do for your clients? And you take these things, you put them on stability, you test, how do you do this?
Well, maybe first of all we can go into what has changed in these revisions.
That's a good place to start. Yeah.
So the current 1207 chapter is 2 pages long. So the revisions expand that chapter drastically. And essentially what the revision of the chapter do is these revisions walk you through understanding your product package system and how that applies to container closure integrity test methods. And that's really what dictates which method you're going to use for your product package system. So for example, if your product is proteinaceous or it's a viscous formulation, you may not want to use a pressure-based test method. Alternatively, if your product is very similar to water and it's not conductive, you're not going to want to use something like high voltage leak detection. You want to expand on that?
Yeah, I think really what the new chapter does is it broadens. It goes from 2 pages to many pages, but the gist of it is to know your product and know, and as Oliver pointed out, use an appropriate analytical method to assess container closure integrity, and then to do that assessment at all stages of the product life cycle. So in development, doing good package development, during process development, making sure that the process that you're using to package this product can in fact maintain container closure integrity, and then also doing routine commercial monitoring during commercial batch manufacture, doing container closure integrity testing in process, and then finally doing container closure integrity testing during your stability protocols, just seeing that container closure integrity is maintained over the shelf life of the product. So it's finding the appropriate technology to do the testing and then doing that testing over all aspects of development and manufacture.
Right, and essentially what you're doing is you're creating an integrity profile of your package, and you're understanding the inherent integrity of your package as well as the integrity of your package post-distribution or during production or or on stability samples.
Yeah, so what the chapter in general is supposed to be doing is first basically asking everybody that deals with their container closure, packaging, and systems and processes related to that, is to take a holistic look at it. To start off with, you start from the beginning. You have to select the right containers and closures that are going to be appropriate for the type of product that you've got. You have to have them appropriately measured. You have to have your suppliers in place, and then you work that into your process. Do you have the appropriate process to be able to handle those containers, close them appropriately, seal them if need be on top of the closing process, and then move that into the filling process? Is there any impact on your process if you have any sterilization impact on your package? And then also, not only is the package going out to clients, some of that goes on stability to determine shelf life. So all of that holistic look has to have some analytical measurement built into each phase of that life cycle of the product. And that analytical piece is an important part, because you can't prove that you have assurance of what your intention is for that package integrity without measuring it appropriately. And that's where the different types of instrumentation that are available can provide some more consistency and reliability for that type of measurement.
I think that's a good point. What the chapter is really trying to do is encourage people to use, I think, more quantitative analytical methods in order to do this type of package integrity assessment. So, Oliver represents a company that covers really two of the three main leak detection technologies that are out there: high voltage leak detection and vacuum decay. And then a company that I work with, Lighthouse Instruments, covers really the third area, doing laser headspace analysis. So between those three analytical methods, you can pretty much cover, I think, any integrity test that needs to be done to qualify a package.
Why don't we go into what sort of an output we're looking at then, Oliver? Why don't we start with you? So,
In terms of output of what?
from the instrument. Yeah.
From the instrument itself.
Yeah.
So,
how do you set a spec? You know what I'm saying?
Yeah, exactly. So a leak test method, again, we're trying to take a known good product or an assumed good product, test it to make sure that the package integrity has been maintained. So you're going to put it in a situation, you're going to treat it a certain way, and look for a certain outcome. So we'll say vacuum decay. Vacuum decay is a method where you place your container in a test chamber or a controlled test space, and you pull vacuum, you monitor the vacuum level, and you look for any change in the pressure. I mean, very minute changes. So if air is leaking out of that package, or if moisture is vaporizing and leaking out, the pressure will change, and we're measuring that inherent pressure change in the test chamber. And so we typically get 2 or 3 data outputs, the actual vacuum level in the chamber, the change in pressure. These are quantitative numbers that you can actually then verify. You can go back, you can retest the package because it's a non-destructive test. You can compare that test result of a package to a master sample that we know doesn't leak. We can introduce positive controls of very specific leak sizes or flow rates and say that that is the target leak spec, and so we know exactly where to define or draw a line in the test parameters to identify what is known as good and what's defective. So you can apply a lot of traceable standards when you're doing a test method via a quantitative, non-destructive approach. If you're doing a destructive test method or a method that has attribute test data, well, if it's destructive, once you test it, that information, that test has been expunged. You've now exhausted your information. It's either pass or fail. What comes of that, and how bad of a pass or how bad of a fail or how close it was to your spec limits, you'll never know. There is no spec limit. It either has blue dye in it or it doesn't. So it really becomes, if you have a destructive method or a subjective method, you're more in it's a vast gray area of information. But if you're dealing with a quantitative test method or something like Lighthouse Analytical Labs produces test systems that output the oxygen headspace, that's a number, that's a quantitative figure, and you can trace that or track it to standards with known headspace concentrations and say that that's the standard. You have something that's traceable. You have something that you can rely on.
And these tests are conducted at room temperature? The tests are at room temperature, or?
Oh, just about it. You know, any type of environment you can imagine. We have requirements that range from, today I had requirements ranging from -80 degrees Celsius to, you know, sort of, you know, production environments where, you know, food production environments. So these tests can be under a range of conditions, under a range of circumstances. Packaging today and package delivery systems are basically designed to bring a product to clients under a variety of circumstances, whether it's a lyo product, liquid fill, whether it's a cold chain transport product. You know, whatever is involved in how this product will reach the end user, that's really how you're going to be handed the application and asked to assure the integrity of that product. So the circumstances are vast.
And how about this headspace?
Yeah, I think coming back to your question about setting specs,
right?
Typically, and I think that Don can probably comment on this as well. Your specifications for a particular formulation are coming from your formulation scientist. So I think it all starts with knowing your product. Is it oxygen sensitive? Is it going to be packaged under vacuum? Is it moisture sensitive? So all of those product specifications are coming from formulation scientists at a pharmaceutical company. And then those conditions need to be maintained by the package. So if the oxygen level has to be below some percent, the package has to be able to maintain oxygen below that percent. If the package needs to maintain vacuum at a certain level. And then all of these methods that we're talking about, vacuum decay, high voltage, laser headspace, can make quantitative measurements of those quantities in the headspace and demonstrate scientifically that the package is, in fact, keeping oxygen levels where they're supposed to be or vacuum levels where they're supposed to be.
Well, I mean, so your set of specifications, some of it's based on developmental history. The other one has to be really set on what your process experience has been. So is this stuff also used to control a process? I mean, it's used as in-process testing?
Yes. Right. So again, I think coming back to what I mentioned early on, it's really a very holistic process where you start in development, specifications are set, you're testing packages to see that they can meet those specifications. Then you're moving into process development and showing that your process can manufacture packages with those specifications intact. And then your commercial batches. Some people are doing 100% inspection of things that are perhaps higher risk than others. Other people are doing statistical sampling of commercial batches to demonstrate that their processes are being maintained under control. And then the final part is the shelf life testing.
Can we, I just wanted to get some idea what the high voltage measure. So what are we doing there? Why don't you explain what that's?
So high voltage is basically, it's treating the package as a resistor. So we're applying high voltage to one side of the package and measuring for, or offering a ground current source or a position on the other side of the package, and we're measuring that through current if in fact there is a defect or not. So basically the package acts as a resistor. The package materials are non-conductive. The liquid contents of the vial or package would be conductive. And so if the package has maintained integrity and you have you know, a complete glass wall around the vial, that glass will not conduct the voltage, and no voltage will actually transmit through the vial. But the moment you have a micro leak, and a very small micro leak, you're basically cutting the resistance of that vial by almost 50%. That barrier, the resistance of that barrier is lost, and the voltage is now able to arc through the vial, and that current is measured.
It would seem to me it's very package specific. So how easy is it to change from different put-up types? I mean, a multi-use vial or an ampule, I mean, or is it just limited to a specific, you understand what I'm saying?
Yeah, very simply put, the package must be non-conductive. Package materials, the product must be conductive, and at that point, the offline system that we offer is actually designed such that it just has to have a rotary symmetry, so you can rotate the vial or syringe, and the high voltage probe and the ground probe will contour that package and apply the voltage along the package. And so it's actually quite transferable from one package format to another. It's if you have a LIO product or other unique package characteristics, that can be limiting. Certainly vacuum decay has more specifications around the package and product, and there we have a package-specific test chamber typically.
And all of these are non-destructive. So in other words, my sample remains intact, so I can go back and retest.
Yeah, all test methods that we're offering, as well as GIMS, they're all non-destructive and quantitative test methods. That's fundamentally what they are.
Okay, I got that. So far,
It's
so
like you
good.
do dynamic studies, so you can measure the same package over a period of time and see how things are changing in order to measure leak rates or measure stability profiles of products.
Okay, so how does your system measure the change in headspace then?
So the way that our system's working, in laser spectroscopy, you shine laser light through the headspace of a container, and so our requirement is that it be an optically transparent package. So plastics or glass are typical materials that we test. And then lasers can be tuned to detect different molecules. So we have systems that measure oxygen or moisture or carbon dioxide primarily. But it's shining laser light through, and then those gas molecules in the headspace of a container will absorb some of that light, and that absorption signal is proportional to the concentration of gas inside of a package.
Doesn't it assume then there's some sort of outgassing from the solution? No, I'm not following it.
No, that can happen. Typically, say, for a liquid product that is oxygen sensitive, it comes through the filling line, it's dosed with the liquid, and then it's purged with nitrogen in a second station, and then it is stoppered and then capped. And then you can non-destructively measure the oxygen content in the headspace. So there may be some outgassing from the solution over time, but that's an important thing to understand.
Okay, but
as well.
that would you develop that as part of the background for your product. All right, Justine. So how much? So let's talk about method development for these applications. There, how much is involved?
Sure, actually method development is extremely important for these CCI methods. Every CCI method must be developed specific to one product package system. So for example, if you have a vial that has two different products in there, say you have a LIO product or a proteinaceous liquid product, you may want to explore two different types of methods. So again, it all goes back to understanding your product, your package system, and what your study goals are for that method. So again, so if you need to optimize your method. So for example, for vacuum decay, the test unit is going to be encased in a test chamber that's specifically created to hug the outer contours of that package. Because vacuum decay is highly dependent on the volume within that chamber and also the volume in the tubing or in the entire system. So vacuum decay consists of several testing steps, a certain amount of seconds to evacuate the chamber, to pull that vacuum. Typically for a glass vial or for a liquid product, you would pull a 1 atmosphere differential. If you have a LIO product, you might not want to pull that much of a vacuum, but those steps need to be optimized utilizing sets of control groups. And that's really what 1207 goes into in detail, the types of control groups that you need to utilize when developing your method. So you have one set of negative controls, and these are your non-leaking intact packages from which you establish your testing baseline. So these methods give you a quantitative value. Those negative control samples, that system baseline, needs to fall within those within a certain range of those quantitative values. Your second set of controls are your positive controls, and these are packages with an intentionally made defect. So for example, there are laser drill defects. These defects are created in the actual component material of your sample. So if you have a vial, you would want to create the laser drill defect in the vial wall. And these laser drill defects can be created to a certain micron size, 2, 5, 10 microns. You want to create laser drill defects or any defects in a range of sizes to build in that linearity of your method. It's very similar to any analytical method. I think that's what we really want to stress, is that, you know, CCI is not an art form, it's a science. It's an analytical method that needs to be developed and optimized.
So you come up with a calibration chart, is what you can.
Essentially, you're coming up with a range for your non-leaking packages to fall within. And any package that falls out of that scope, out of that range, is considered leaking.
Is that all true for the headspace as well?
It is. I think to expand a little bit on Justine's comment, so all of the analytical methods that we're talking about here are calibrated in their own particular way. And then after an instrument is calibrated and demonstrated to be making accurate and precise and linear measurements, then you use these positive and negative controls as part of your experimental method, along with testing a bunch of samples of product, in order to demonstrate that with the positive controls that in fact the change you're expecting to happen happens, with the negative controls that nothing is happening over the test period, and then your product samples are the ones that you're looking to see if in fact anything is happening during the test.
Don, is this how you use this in practice, or how do you do this?
Yeah, well, the Chapter 1207 is meant to be a guidance chapter. It's not meant to be an inspectional or compliance aspect to be compared against, or to be found someone's doing something wrong. It's basically supposed to provide the information for best practices of different types of methodology and how to approach container closure integrity from development all the way through release of product and shelf life. Thus, there's a lot of different aspects in there, and as long as everyone realizes that the information is there to use for their specific purposes, their case-by-case use for products, then it's going to be a very useful chapter. It intends to tell everyone that there's no one-size methodology that fits all containers and all products. It's a very rare case that someone out there has one product and one container that they have to be concerned about for container integrity, and thus only have to select one method to use for that. But it's very possible that even that one container that someone might have may require different methodologies through the life cycle to determine what types of questions that they're trying to answer. And thus, as I said, the chapter is meant to provide that information in a very broad approach, and provide information that hasn't been available to a lot of people other than packaging engineers for a long time. And now it's out there for a lot of people to learn what package integrity is about, what leaks and leakage really mean, with the difference between a leak and permeation, the difference between a destructive test and a non-destructive test, and the fact that there's different types of analytical methodologies. Some are very sensitive, but are sensitive for a specific reason, and some are less sensitive, but may be very applicable and appropriate for different purposes. Thus, it's not a chapter that's supposed to say, here's the method for this particular type product container. It's supposed to be able to say, here are your options, select those that are appropriate, and here's the information that you can utilize to take you a few more steps further to learn that information for your product.
So are we limited to only glass? I mean, I got a sense based on what you're saying is you can use polymer as well. You could have a polymer bottle. Okay, so you could use this to detect leakage in an oral liquid. Is that right? I mean your instruments. I understand that there's the guidance and then there's the hardware. I'm just, because to me, I'm thinking if I was making a suspension or something that's liable to be it's susceptible to that. So does this apply? Can you apply this technology? What do you think, Justin? Does that not come?
You can certainly apply this technology, or all of these technologies, to different packages of different materials of composition. We've explored plastic syringes, plastic vials, but there are other different considerations that need to be considered when you're testing a plastic vial versus a glass one. As Don mentioned, permeation may be an issue. For example, if you're exploring helium leak detection, if you're testing a plastic vial, helium permeation through that material is going to be much more of an issue than if you were going to test a glass vial, of course. So that needs to be considered when developing your method or when choosing your method.
There's a lot of complexity coming out in the pharmaceutical industry today, and a lot of different packages are being looked at for a lot of different products to be able to support the integrity of those products during their shelf life and before they get to the customer. And there's also complexity with the combination of medical devices along with standard containers that have been in the pharmaceutical industry for a long time, such as pen injectors and different types of applications like that. And these are adding complexity to the approach to doing container closure integrity and challenges as well. And because of that, all of the suppliers of the standard instruments, as well as some new ideas which are making modifications to those standard instruments, as well as other types of methodologies are out there that haven't been proven yet, but are being tested and in the developmental stages are being looked at, because we're starting to add complexity and starting to add different types of packages, such as the plastic single-use bags. There is no standard methodology out there that is yet to be commonly used, although there are some that are being used a lot. And eventually, if they are being found to be analytically reliable, as well as consistent and robust, then some of those technologies may come out to the forefront for that particular type of package to be able to be used.
And also, I mean, just to be clear, all CCIT and package integrity testing is a risk-based approach. Any container testing observed by the FDA would be on a risk-based approach. And so, you know, parenterals, they're at the forefront of risk. That's where all the development is. That's where the unique applications are requiring the highest level of assurance. And that's where the new test methodology is being developed and the new methods are being made available. And so really, when we're talking about USP 1207, this is the forefront of container closure integrity testing. All these methods that are listed in that chapter can be applied across the board for a variety of other pharmaceuticals, including, you know, oral dose, lower risk applications, or applications where the efficacy of the drug is reduced by the presence of oxygen. These sort of things. It may not be super high risk, but you certainly can apply one of these test methods too.
Well, I think the comment Don made about the injector pen, it seems to me inherently complicated. I mean, so how do you even go about that? Has anybody done any of that? I mean,
yeah, there's a few ways to do it.
It sounds like a nightmare to me, but go ahead.
Who wants to tackle that one? Yeah, go ahead.
It's certainly an up-and-coming issue right now. We see so many more auto-injectors on the market in recent years, and so what Whitehouse Labs has done is, for our clients, we recommend a two-pronged approach. So for auto-injectors, we want to test the device with the syringe in place, so the assembled device. So for that type of package, we would go with a pressure-based technology. However, this typically runs the risk of that formulation clogging defects. However, that is really the only technology right now that can be utilized to test the entire device, to have 100% inspection of that particular sample.
Do you have to take the device apart to test it?
No, you don't. That is an advantage to using a pressure-based technology such as vacuum decay for testing an actual assembled auto-injector device is that you don't have to disassemble it. However, what if you run into that issue where your product does clog defects? So I mentioned earlier the positive controls with tiny micro leaking defects. You essentially won't be able to detect those leaks due to the issues with product clogging for a pressure-based system. So I mentioned a two-pronged approach, that vacuum decay or another pressure-based system such as mass extraction would be the first prong, and then the second one would be just to evaluate the pre-filled syringe on its own.
So by clogging, you're talking about self-sealing?
I'm talking about the product formulation drying around those intentionally made defects and blocking that pathway for the liquid to be able to be volatilized through that defect due to the vacuum that's being pulled on the product. So that's a real issue right now for liquid products.
And it's a very good question, and a very good question that may lead to better thinking across industry, especially with suppliers, and the fact that because we're getting into more complicated packaging industry in general has not really taken into account the fact that the integrity testing is one of the key analytical pieces of assurance, and especially from the sterility assurance aspect, let alone just product assurance and product quality. Thus, if that's taken into account in the designing of these, and there's a lot of new packages out there that are designed specifically, especially pen injectors designed specifically for some products, then why not take into account the fact that they need to be analyzed and asked, across industry, what are the types of analytical methods out there, and what do those analytical methods require in order to be done appropriately, and can we design a package that can be both an easy-to-use package or pen injector as an example for the patient, as well as being analytically easy to test with the type of equipment that we already have out there. And that question hasn't been asked.
It just seems to me that's the physics of what happens. They are gonna be self-sealing. I never thought about that, but so there's got to be some augmentation of that. In other words, to me, if it's gonna self-seal, you either got to compensate by pressure, or you got to compensate by, putting it into a lake. I mean, the way this stuff used to be done, you put it into a dye bath and pressurize it, and then throw everything out. Right? I mean, that's pretty much it.
You wouldn't find any defects that way.
Yeah, but that's what they used to do.
All of your packages would pass.
And today, one of the technologies that I think that is sort of curtailing that circumstance is high voltage leak testing, whereby you're not really requiring any transfer of the product or dye or any sort of mass. We're literally just transferring electrons through the product. And so, when that voltage arcs through the vial and the product, that's the only thing that needs to get through the defect, and that's it.
I got you, Oliver, but to me, see, I was going to do that again. So to me, how do you do that with a pen injector? You'd have to take it apart, no?
Yeah, would have to take it apart. You could potentially the pen injector has windows present where you can present the probes for the test. But if not, then you're really subjected to doing a vacuum decay test. We have encountered products as well where the product itself is non-conductive, so that rules out high voltage. The product itself plugs up defects, that rules out vacuum decay. And then you're left with only a very few leak test methods left, let alone any that might be quantitative or reliable. And so when you're faced with a challenge like that, you have to get creative, but also within the scope of what's required. So you could test the auto injector or the product with not active ingredient to validate the system, and then resort to another method that may reduce the level of assurance on the actual active ingredient. But
I got you. That sounds like an opening shot here for the headspace then, huh?
Well, It is, and again, it comes back to knowing your product and the package, and particularly for headspace analysis, if there is optical access to the parts of the package you're trying to measure, then and that headspace is modified in some way, then the laser headspace method is applicable to assessing container closure integrity. So again, it comes back to no two packages are alike, no two products are alike, and you really need to look at both, understand what it is you're trying to measure, and then go find an analytical method that can do the measurement.
So what has been generally, I mean, everybody will get the chance to answer in kind, but I mean, generally speaking, what's the go-to technique? I got to believe that there's a preference.
There is none.
There isn't.
There isn't any.
It's
then,
highly dependent on your product and your package.
As I said, when we started to rewrite the chapter for 1207 in the USP, the intent was to show that there's a number of different methods to choose from. There is no one method that everyone can decide to go to because someone's telling them that's the best method out there, because there is no best method for all packages. The idea basically is you've got to make your own decisions. You got to make decisions based on the beginning of your design of your package, on what it is you're trying to prove, what it is you're trying to assure of the product quality, and then find the appropriate methodology that'll be able to meet that requirement for the package that you've got.
But I do think that probably the only answer or correct answer to your question would be a method that's quantitative, ideally non-destructive, but a method that's quantitative and reliable. If you have those ingredients to a test method, and you're going to have a test method. If you have a method that's probabilistic, you're really left to chance. So.
Well, to me personally, coming from the oral solids area, to me, Nirvana was any time I could test something and I had it and it stayed there. So I mean, other than measuring it, we destroy the stuff. Mm-hmm. But what we found was, by getting creative, we found that you can use interferential holography. We would actually get a refringence pattern from these things. But again, the problem with that was everything was unique. Everything was it became very, very time intensive. And the question really is how much is that really going to tell me to tie it back into my process? So to me, why do I say that? Anything that's non-destructive's got my vote. So to me, I'd rather be able to go back and test it again.
Something that's also, I would say, quite dangerous is that there's a lot of test methods that are out there in the field right now that are, you would say, sort of a standard. So the water bath bubble test, you find it across the packaging industry. You put a package in the water bath, you look for bubbles at half an atmosphere. So it's sort of a standard to always go to half an atmosphere. And many people do it without validating the method or proving that it works, and they just roll it out as a grandfathered test method. And you're certainly not going to have a reliable, repeatable, or a consistent method if you just roll out a standard. So like Jim said, there's no one method that's go-to, and every package is different, and therefore you really have to understand the package and the product and apply a method accordingly.
I will get calls constantly from someone in the industry asking to do some testing, and once I ask a series of questions, realize this is not a problem that we can solve with our technology, and we'll give them Oliver's phone number. And I've received as many phone calls from people who have just gotten off the phone with Oliver saying, hey, you should talk to Jim Veale at Lighthouse. So there's no one method that does everything. And I think that this is one of the hopes of the new chapter 1207 is to give people in the industry a better overview of the current quantitative analytical methods that are available to be used. But there is going to be, I think, and is continuing to be a big educational effort across the industry to make people aware of what's applicable and what's not applicable.
So Don, were you involved in the rewrite? Are you part of the team that did the rewrite?
I was part of the USP team that got together to make the revision of that chapter, yes. There was an expert panel that was brought together from the USP, and I was part of that panel. one thing to say about the different types of methods, and you know, I do appreciate the fact, because I'm an analytical microbiologist, and quantitation is important to most analytical chemists. In microbiology, it's important but not as critical, because it's very difficult to quantitate microorganisms because of the variability aspect. But there's a lot of energy, I should say, put into some of the comments that we've gotten back from the chapter in order to move that chapter and progress it forward with the feedback that we've gotten from the public. And some of the comments had to do with the concern of, number one, not understanding the difference between probabilistic and deterministic methods, because we brought that definition into the chapter. And also the thought that the chapter was intending to encourage people to use deterministic methodology over probabilistic methodology, and that was not the intent of the chapter. The chapter was to explain which types of methods that do exist today, that are being commonly used, that are in either category. And there's a time and a reason for using the different methods for different points of the life cycle of the process. A lot of people have used dye intrusion, a lot of people have used microbial immersion, and there's different packages and different points in the life cycle of a product where that type of information can be very useful for testing the integrity of a product. And people have chosen to do that, and the regulators are learning along the way where those appropriate points in the life cycle of a product that they may be useful. When it comes to quantitation, if you need a point in the process where you need quantitation, then you can't use a probabilistic method. But if you only need a qualitative, does it pass, does it fail, you can use a probabilistic method and it works fine.
So my question is, it sounds to me like you got to know an awful lot about whatever the intrusive or extrusive, either going in or coming out, what the chemistry is of that before you can use either method. I mean, otherwise, so it leaks. Great, okay, so what, right? I mean, the answer is in risk, there is what's the chances, what's the severity, and what is the, well, so the question is, you got to have a really thorough, I would think, chemistry. You have to have a pretty good idea of the chemistry of what's going on. Am I right? I mean, it's not just microbiological contamination, I'm thinking it's degradation, right?
Well, it's stability and sterility of the products. So sterile products obviously have to have a microbiological barrier, but many of these sterile injectable products are also highly oxygen sensitive or moisture sensitive, and the packages need to keep those atmospheric reactive gases out of the packages as well.
Yeah. So there, yeah, as Jim mentioned, there's a very important balance to make sure that you understand your products. And actually, the approach that industry has taken, where to develop a better understanding of the product through you know, the design aspect, and really doing as much upfront information gathering and learning of your product, you know, as you're working it through the process and trying to ensure the quality. You have to understand your product formulation, you know, and what things can affect that formulation. And you're right, it's a sterile product, so you're going to worry about microbial contamination. But microbial contamination may only be one part of that. It's not the sole part. Thus, when you try to select a method that's quantitative, as an example, for package integrity, you're going to want to know what's the intent of that method. Are you trying to show just that it's able to maintain sterility over the life cycle of the product, especially over stability, or are you also trying to prevent other contaminants from getting in there, such as oxygen, as Jim mentioned, because it doesn't matter if it's not sterile if the product is inefficacious anyways. You know, you've got 2 hits against you,
Yeah.
and it doesn't matter if a very tiny leak will not allow a microorganism in, but allows oxygen to get in, and oxygen is going to degrade the product, then you're lost anyways.
I agree. So Justine, let me ask you before, so how solid of a package do your clients come in with? Is this something you develop? Is this something that you advise them? Do they give you the information? What's the deal?
It really depends on what phase of the life cycle they're in. If they're in the design phase, that is up to us to determine, and or typically we can establish maybe optimal capping setting for your vial in that regard, or we can use a method that is very, very sensitive to enable us to conclude if your that your package components even fit together properly, or if they can even seal properly. In that regard, yes, we would be able to determine that for our client. But later on in the life cycle in production, and also when you're on stability, we'll utilize these methods as more of a verification check.
So most of these things are incorporated as not only an end test, but also as a stability indicator or stuff like that. So
not really a stability indicator. Essentially, what we'll do is we'll utilize these methods as a stability check that the product itself hasn't affected the package. So not really the stability of the product, but the package system.
Okay. All right, so we're gonna kind of wrap this up here. So other than the fact that now we know about the guidelines and we know that there's several tools out there that we can use and not one size fits all, okay, we got that down. Is there anything else that you guys want to leave the folks with tonight? Or are we all set? Any one last comment? Go ahead, Don.
I'd just like to add that there's the USP and the PDA have been working together over the years, especially in the package integrity area. And there's a technical document that's that exists right now, TR27, which is an old and outdated document, but it still has some useful information in it about pharmaceutical package integrity testing. That document is undergoing revision now, and there's a committee including a large number of people, including some of those at a panel with me here. And what that document's going to be in the future is a complementary technical document to the USP chapter. The USP chapter is a guideline and a general information to what kind of testing there is out there and what are the common approaches in understanding package integrity. The TR27 from the PDA is going to be a challenges and issues type document to talk about what are the issues people have with package integrity testing today, with the types of packages that they have presently and potentially in the future, and what are the challenges and approaches that other people have taken to approach that. So we'll be able to talk a little bit about what is the upcoming and current technologies that are out there that are kind of breaking the barriers for the issues that people have with the difficulties in package integrity testing.
So how would somebody get involved with something like that?
There is a committee already set up that's been in place for the past year and a half, and if they just keep in touch with the PDA, if they're a member of the PDA, or if they know someone who's a member, then they'll become aware as the document progresses, when that document goes out for review, and then when it's published, anyone in the PDA will be aware of that document.
Right, and the same thing with the USP 1207 chapter. The initial revisions were released in the Pharmacopeia forum in September, October of last year. There was a comment period that has since elapsed, but the chapter itself is undergoing a second revision due to those comments. And so once those second revisions are released again, there will be another subsequent comment revision. So anyone is able to provide their comments and suggestions and feedback if that's necessary, and you feel that you have something to provide.
Okay. Well,
yeah.
Thank you, Justine. Thanks, gentlemen. Thank you very much for your time. That concludes our session on packaging integrity. Thank you.
Thank you.